So I'm here to talk about our science collaborative project entitled Habitat Heartbeats, incorporating Bivalve Biosensors into estuary monitoring. Our project team includes myself, Luke Miller from San Diego State University and our Coastal Marine Institute, Sarah Giddings from the Scripps Institution of Oceanography, who's our technical lead, then Kristen Goodrich from the Tijuana Estuary National Estuary Research Reserve, who is our collaborative lead, along with Jeff Crooks, also from the Tijuana NERR, who is a project team manager and one of our primary end users. So our project goal here is to try to develop a biosentinel monitoring system, monitoring system we can put on bivalves like oysters and mussels that live in the local estuaries, and ultimately provide real-time data on behavior and physiology for these animals in response to changing environmental conditions. And this is born out of a need of our primary end user, the Tijuana NERR, and the California State Parks, who manage the local estuaries in the San Diego region. And these estuaries are low inflow estuaries, so they don't have a lot of continuous major flows from the terrestrial realm into the ocean, which means that during periods like winter, ocean waves can actually pile up sandbars on the mouths of these estuaries, close them off, and that can lead to hypoxic events and even die-offs within the trapped estuary water. And that then triggers the local managers to make a decision as to whether to literally spend the money to bring tractors in and physically open up these estuaries again. And so it may be useful for those managers to have additional tools that give them some idea of what the biotic system is doing in addition to the water quality parameters within those estuaries. And then we envision secondary users being groups such as aquaculture practitioners, restoration efforts such as living shorelines efforts where oyster reefs are being rebuilt in the local habitats, and then also working with Sea Grant, California Sea Grant, to identify other potential users through their extension program. So what we are doing here in terms of making biosentinels is developing small sensors that we can attach to the outside of bivalves, such as mussels and oysters, that can measure things like the opening and closing of the shell and the heart rate of the animal. In both cases, these are sensors that can be attached to the outer side of the shell, so that there's no invasive sort of surgery needed on these animals. And these sensors can be left in place for months at a time, giving us a fairly durable long-term monitoring solution. And then as part of this project, we will implement a cellular modem system in order to be able to stream at least snippets of this data up into the cloud and make it available in a near real-time fashion. And then ultimately, we are going to sort of do all this using open source tools so that we can release both hardware, software, and data analysis tools to go along with these projects, and those can be things that can be then modified and adapted by other potential end users for their own purposes. So what we are doing is making some medium to long-term deployments of instrumented animals in the local estuaries. Our map here in the lower left shows in red boxes 3 of our targeted sites: Los Penasquitos Lagoon in sort of the mid-San Diego County region, Tijuana Estuary, or the Tijuana River Estuary, down at the southern end of San Diego, right on the US-Mexico border, and then San Diego Bay itself, which has sort of an estuarine system at its southern end. And then all of these systems are fairly urbanized and fairly heavily impacted by human development, both historical and modern day efforts. And we're using a mixture of both moored floating deployments and benthic deployments. So in the mooring system, we're attaching oysters with their instruments floating near the surface and near the benthos of these tidal channels to look at vertical stratification or effects of different halocline, thermocline, et cetera, and oxycline changes in the water column. And then the benthic deployments are really to sort of give us a picture of what happens to these animals as they're also undergoing natural immersion and submersion cycles with the tide every day. So in both cases, we can collect data on when they are open, when they are closed, when their hearts are beating fast, when their hearts are beating slowly, in order to get a picture of how they are responding to their environment. Along with those data collection efforts, we've also implementing a needs assessment and interview process with various other end users being led by Kristen Goodrich. And so we're basically doing structured interviews, getting thoughts on how these potential end users might make use of these sorts of data streams, which of these data streams might be useful for them, and then potentially from their suggestions following it on with other potential users. And along the way, we are bringing these potential users into an advisory group for this project so that we can keep them updated on progress on the data collection process, the development process, and then use them to sort of help us guide what project deliverables might be most useful to them at the end of this project. And ultimately, our goal then is to produce this BioSentinel monitoring system that can provide real-time updates to the cloud, share those designs with users using open source tools so there's low cost entry and the ability to modify and change the system as needed. And also in the process, collecting water quality data and building models for how these animals are responding both behaviorally and physiologically to changes in temperature and salinity and oxygen availability and so forth, and try to then boil those sorts of responses down into perhaps more simplified metrics that managers can use to augment their decision-making processes.